Welcome. In this session, I'm going to start the second year syllabus. You know already in the first phase we completed 50% of the portion. Now we will finish the remaining 50% of a second year portion. Okay. The first chapter which I will be discussing from the second year syllabus will be principles of inheritance and variations. So let's see what this chapter is all about, the principles of inheritance and variation. So this title I can just put it into one word: genetics. Okay, so we are going to study about genetics in this chapter. And what is genetics? So this genetics is study of variations and inheritance of characters. So study of variations and inheritance of characters. So what are these variations? So here variations are differences. So variations are differences between individual members of a species. So differences between individual members of a species. So we know our brothers and our sisters do not look same. Of course we are all humans, but there are variations. So the facial features, okay, the height, weight, okay, the texture of the skin, okay, or the texture of hair, or curly presence of curly hair or smooth hair. So there are so many variations that we come across between siblings, and not only that, individual members of a species also we find the differences between them. So all those differences we call them, okay, variations, right? And we studied in the first year that the meiosis is the one that plays key role in production of these variations. Okay, so there we have studied in a packaging stage that there will be crossing over, and crossing over will result in recombination of genes, and that results in production of variations. Okay, so in the cell division called the meiosis. So in meiosis we studied crossing over takes place, and crossing over results in recombination, and the recombination results in production of variations. Okay, so those are, okay, differences between individual members of a species. Then what is this inheritance? So inheritance is nothing but heredity. So the characters from the parents to offspring. So here the passing of characters from parents to offspring. So that's what we call inheritance. So I can say the transmission of characters from parents to the offspring. So we inherit some characters from our parents. Okay, so some characters from the father and some characters from the mother. So that is what we call heredity. So heredity is inheritance of characters from, okay, parents to the offspring. Okay, so that's about the genetics, which is a study of variations and, okay, inheritance of characters. And we understood what actually these variations, they are the differences between the individual members, and inheritance is nothing but heredity, which is, okay, passing of characters from parents to their offspring. And humans know that these variations, so the variations were present in sexual reproduction, I mean, the cause of these variations is the sexual reproduction. So we don't see, okay, in asexual reproduction, production of these variations. We see only in case of sexual reproduction. Okay, so the sexual reproduction causes variations. The sexual reproduction, of course, it is hidden in the sexual reproduction. We know in the sexual reproduction, meiosis takes place during the formation of gametes. Okay, so that's what I have mentioned here, and that will result in crossing over and the recombination, which finally results in the production of variations. And humans knew that the sexual reproduction is the reason for this from, okay, 8000 to 1000 BC. So during those days only we have come to know, but we were not knowing the scientific reason behind it. Okay, so what people used to do from that time, so they used to select the plants and animals that have desirable characters. So they were doing selective breeding and selecting the plants and animals with the desirable characters. Okay, so that were naturally present in wild populations. So they used to see a particular animal or a plant, say suppose a cow or a cattle that is able to produce more amount of milk, okay, or maybe producing in some case more quantity of meat. So by doing and again selecting the plants that produce bigger fruits or tastier fruits, these are different characters that they have observed, and they started to breed and then produce the new offsprings that have desirable characters. So they used to select plants and animals, okay, by selective breeding. So by selective breeding, of course, this selective breeding, I also call it artificial selection. It's called artificial selection. Okay, then and also selecting by or selecting plants and animals that have desirable characters, means the characters that we require. So those plants are said to be having, or animals having desirable characters, right? And here I will give you an example, right? So artificial selection artificial selection and domestication, by artificial selection and domestication of wild cows we now produced uh, a breed of cow. So we developed a breed of cow. It's called Sahiwal cow, okay, of Punjab. So in Punjab area, we developed this Sahiwal, okay, cow of Punjab. So this Sahiwal cow is, okay, one of the best milch breed, is one of the best milch breed. I hope you already know the milch breeds are very good milk yielders. Okay, so in course of time, right, by artificial selection and by domestication of these wild cows, we have the Sahiwal cow now, and that is one of the best milch breeds in India. Okay, and not only in this case, we have developed in so many types of animals and also in case of plants by using this artificial selection and domestication. Even the different types of dogs what we see, so those are also by artificial selection and domestication. So even in case of horses, we can see there are types of horses. The best horses, the one that are used in racing, okay, and for other purposes. So it is all by domestication and artificial selection of wild species, and resulted in, okay, formation of plants or animals with okay, desirable characters, right? So this is how by artificial selection and domestication of animals or plants, we developed new breeds, okay, new varieties of plants in course of time. Okay, so this is a little information on what actually are those variations and inheritance. So we will be studying scientific reasons behind this. So I told you in earlier days, these people, they know only this artificial selection and domestication, and the reason, that is the scientific reason behind how these characters are inheriting from parents to offspring, all these they were not knowing. And from long back, many scientists they tried, okay, to find out this inheritance of characters, but they failed, right? And the experiments conducted by Gregor John Mendel became successful, so that later I will discuss. So before I start discussing the chapter, I would like to give few terms that come in uh, genetics. So terms in genetics. So first we will try to understand these terms, then later on we will move on, okay, to understand the principles of inheritance and variations. Okay, so here let's see the character or trait. So what is a character or trait? So this is a well defined morphological, so this is a well defined morphological or physiological feature of an organism. So it's a well defined morphological or physiological feature, okay, of an organism. So that's what we call trait, like the skin color. So we can, if I take in case of human beings, we can take, okay, skin color, okay, then hair texture you can take in case of human beings. Then if I take in case of plants, okay, the seed color, okay, or flower color, etc. Like this there will be so many examples you can get. So that is a character or trait. Okay, so usually we use the word trait in genetics. So it's a well defined morphological or physiological feature of an organism is trait. The next one, we'll see what actually is this phenotype. So I will be frequently using this word, okay, phenotype, in this chapter. So phenotype is the external appearance, of an organism. The external appearance of an organism, that's, okay, phenotype. So how it is seen to outside is the phenotype. And one more word, this also we will be coming very frequently, okay, genotype. So this genotype is the genetic makeup of an individual. So genetic makeup of an individual. So means what genes are present in an organism. So what genes are present inside me, so what genes are present inside me or in someone else. So this genetic makeup of an individual cannot be seen to outside, so we cannot come to know what genes are present inside a person or inside an animal or inside a plant. That is not possible, but the phenotype we can see it. So the phenotype is the external appearance of an organism. All right. Then we already know about haploid and diploid conditions. So haploid, so we'll represent with N. So haploid condition, which means uh, a cell that has a cell that has okay one set of chromosomes. So if a cell that has one set of chromosomes, then the condition is called haploid. And coming to diploid, that is 2n, so is a cell that has two sets of chromosomes, has two sets of chromosomes. Say like example in case of human beings. So okay, 2n, that is diploid condition, is 46 chromosomes. So in these 46 chromosomes there are actually two sets. So 23 chromosomes form one set and the remaining 23 chromosomes form okay one set. So you can see here there are two sets. So that is the reason why we have set it as diploid. So diploid means okay a cell that have two sets of chromosomes. So here in case of human beings, every cell will have two sets of chromosomes. Okay, so of course that diploid cells will undergo meiosis and produce gametes. Gametes are the one who will have only one set of chromosomes. So I can say here example, the gametes. So gametes will have only one set of chromosomes, whereas diploid means the somatic cells. Somatic cells means all the cells that make up the body of an organism. So they are somatic cells. So these somatic cells will have okay two sets of chromosomes, right? Of course there may be exceptions. So here, as I have taken in case of human beings as an example, so I'm just mentioning with that only. Okay, right. So we understood that the diploid condition, okay, what is a diploid condition? Having two sets of chromosomes, and haploid that has one set of chromosomes. So every cell of us is having two sets of chromosomes. Of course, this one set will come from okay one of the parents, so maybe from male or female parent. So say suppose this one set have okay from father, and this one side will be from mother, right? So every diploid individual will inherit okay two sets. So one set will be from one of the parents, so maybe from one set from male parent and another set from the female parent. Okay, so that's about a haploid and diploid conditions. Then coming to an another term called allele. So what is allele? Okay, so this allele I can also call as allelomorph is another term. Allele or allelomorph are one and the same. Now what is this? An allele is one of the two alternative forms, is one of the two alternative forms of a right character. So it will be one of the two alternative forms of a character. Like for example, if I take seed color, so in case of this seed color, we find, okay, say yellow color, so some seeds which are yellow in color, and some seeds which are, okay, green color. Okay, so seed color may be yellow and green. So this yellow is one allele and this is another allele. Okay, so of course here I just give a little idea on allele. Don't worry if you do not understand. Also later in our coming sessions I'm going to discuss very detailedly about this allele. So how alleles are formed, okay, how and when we can call, okay, a particular gene, okay, as allele. Of course allele is nothing but a gene. Those things we will see, okay, in our coming sessions, right? So the next homozygous. So I will be again using this word very frequently. What actually is this homozygous? And so here homozygous means an organism. So an organism, okay, having two similar genes for a particular character. So here organism having two similar genes for a particular character. Okay, so we will take the same seed color. So just now I mentioned, okay, in case of allele, so we'll take seed color. So in case of seed color, I told you there is an yellow color, okay, and green color. Now I'm going to mention this yellow color, okay, with the capital Y. I'm going to mention this yellow color with the capital Y, okay, and this green color I'm going to mention with the small y. So don't worry, later we will understand those things. So say suppose this color I mentioned with the, okay, capital Y, and this one with the small y, the green color. Now right, in an organism, so especially in case of diploid organisms, okay, so there will be, okay, right, two alleles for a character. So for any character you take, there will be two alleles in case of diploid organism. So if you are considering a diploid, okay, organism, any organism which is diploid, okay, so in that case we need to take capital Y and capital Y. So you can see here both, okay, alleles are of same type. If it is the case, then this is the condition, we call it homozygous condition. It's called homozygous condition. Or you can take a small y, okay, small y, small y. This is also homozygous condition, right? But if you take one capital and one small, then it becomes heterozygous condition. Okay, so first we'll understand this. Homozygous means organism having two similar genes for a particular character. So for this character, okay, I mean the seed color, it has, okay, two similar genes, that is yellow color. So this is yellow color, this is also, so same genes are present or same alleles are present, and that condition we call it homozygous condition. Okay, it's called homozygous. Right, now we'll see about heterozygous So here, in case of heterozygous organism having both dominant and recessive character, or organism having, okay, two different genes for a particular character. So organism having two different genes for a particular character is what we call the heterozygous. So I'll take the same example here. So in this case, okay, again the diploid organism. So in this I'm going to take capital Y, small y. So you can see here there are two different genes for a particular character. So for that reason we need to use the word heterozygous. So this is a heterozygous condition. So whereas these two conditions are homozygous conditions. Okay, right. So of course, if you take this as a dominant, so usually in case of genetics, the dominant character will be represented with the capital letter, and the recessive character will be represented with the small letter. So this is recessive, and you need to take same alphabet for a particular character. So for seed color, so I have taken Y alphabet, in which you need to mention the dominant with the capital and recessive with the small letter. So that's a rule that we need to follow. Okay, right. So here both are dominant, both are recessive, then the condition is homozygous. But you see in a person, okay, or in the plant, so here we have taken in case of plant. So here in a plant, so that has a dominant gene and also recessive gene for that character, then the condition becomes heterozygous. Okay, so again, don't think that an organism will have all homozygous and all heterozygous. So that will be not the situation. So if I give me as an example, so in me I will have homozygous conditions for some characters and heterozygous for some characters. So like that, every organism will have homozygous as well as heterozygous conditions. Not just for all the characters, you will not see either homozygous conditions or only heterozygous. It is not. So an individual may have homozygous as well as heterozygous conditions. Okay, so that's about the homozygous condition and heterozygous condition. Then the next one we'll see about pure line. So next we'll see about pure line. So I can also call it a pure breeding line, also called, okay, pure breeding line. So what is this pure line? So this will be a strain of individuals homozygous for all genes being considered. Okay, so a strain of individuals, okay, having homozygous condition, individuals having homozygous conditions. So that is it will have homozygous genes, having homozygous, okay, for all genes considered. So a strain of individuals having homozygous for all genes means whatever that characters we are considering, for those characters we see everything homozygous, like we have seen in case of earlier, okay, seed color. So I'm taking this character seed color, and for this it has capital Y, or small y. I told you both these are homozygous conditions. So a plant or animal that has, okay, all homozygous genes, okay, for that particular characters, then we say that as a pure line, is a pure line. Okay, uh, and these pure lines are produced by, so pure lines are produced by continuous, okay, self pollination or self crossing. So they produce these pure lines by continuous self pollination. Okay, say suppose a flower that is bisexual, so I'm going to take the pollen grains, okay, from this okay, anther of the same flower, and I'm going to put that on the stigma and allow it to undergo, of course, fertilization and everything, and then I get seeds. Okay, and those seeds I'm going to raise, okay, into, right, different plants, and again on this plant you have a flower. Again the pollen of that flower will be put on the stigma of the same flower. And so this is first generation, okay, say this is the first generation, and then again the seeds I obtained from this first generation, I use it to raise the second generation, and again in the second generation I'm going to do the same process, okay, going for number of times self pollination. I hope you already know what actually is the self pollination, cross pollination, that you have studied in sexual reproduction in flowering plants. Okay, so pure lines are produced by self pollination or self, okay, crossing, self-pollination or self-crossing for a number of generations, so that the genes for a particular character will come to homozygous condition. So that's what I mentioned here. So after continuous pollination, or self-crossing for number of generations, okay, the genes will come either to, okay, both the genes will be in dominant condition or will be in heterozygous condition. Okay, so if it is the case, then such an individual is what we call a pure line. We'll slowly understand as we go on. You will understand what actually the pure line, okay, how we are going to produce those pure line as we see different experiments in course of time, right? So here the foundation for this, okay, branch of biology, that's called genetics, it was actually laid by Gregor John Mendel. So the father of genetics is Gregor John Mendel. Okay, so he is the first person to study scientifically and become successful in understanding how the genes are being inherited from the parents to offspring. Okay, of course, earlier scientists also, they have done so many experiments, but they tried, and I mean experiments in trying to understand this inheritance of characters and so, but they did not become successful, so all their experiments were failed. But he's the person, okay, whose experiments became successful and made us to understand how the characters will inherit from the parents to offspring. So hence, okay, he is regarded as father of genetics. Okay, Gregor John Mendel. So will be from 1822 to 1884. Okay, right. So born in 1822 and died in 1884. So I'll just mention a few brief information on Gregor John Mendel. In short, usually we write as G.J. Okay, Mendel. Right, he was born on 22 July 1822. Okay, so he was born on 22 July 1822. Okay, in Austria. In Austria, and he died on 6th January 1884 in Austria. Right, and he selected. So he selected a garden pea plant for his experiments. He selected a garden pea plant for his experiments, and that garden pea plant scientifically is called Pisum sativum. Okay, so this is the garden pea plant. Okay, later I will discuss why and what is the reason behind selecting that pea plant only. Okay, and how he became successful in his experiments. Okay, those things I'll discuss it later. So he selected garden pea plant for his experiment, and its scientific name is Pisum sativum, right? And he conducted breeding experiments. He conducted breeding experiments on garden pea plant. Okay, for a span of 7 years. For a span of 7 years. So that will be from 1856 to 1863. So that is 7 years. So for 7 years he was doing experiments on this garden pea plant, and after completing his experiments he written a paper that titled Experiments on Plant Hybridization. So the his paper title was Experiments in Plant hybridization. So experiments right, I'm sorry, on plant hybridization. So it was the title of his paper, and this was published in fourth volume. So it was published in the fourth volume of annual proceedings annual proceedings of natural of Natural History Society of Brunn. Natural History Society of Brunn. Okay, so that is the title of the book, Annual Proceedings of Natural History Society of Brunn. It is the title of the book. In this book, Mendel's paper that was titled Experiments on Plant Hybridization was published, and this was in the year in 1865. So in the year 1865. So here one point, whatever the years I have mentioned, they are very important for our competitive examination need. So you need to remember in which year he published, and sometimes they may ask even in which volume, or in what is the title of that volume, and what is the title of his paper also can be objective questions. So you need to remember all those things, and very important they are all. Okay, right. So that's about okay, Mendel. So of course, while he was working, right, and then when he published all his papers and all, so none of the scientific community paid attention to his papers. So no one have taken it seriously about these experiments. Okay, only after several years after his death, they came to know what actually Mendel did, right? So anyway, right, and Mendel's work was not identified when he was alive, right? So here, after his death, 16 years later in 1900, okay, in 1900, three scientists, okay, rediscovered Mendel laws independently. So these three scientists did not do those experiments together. So they have done independently, separately, right? And they came to know that whatever their findings are similar to the findings of Mendel. Okay, so independently, and there are three botanists. Okay, so one is Hugo de Vries. So one person is Hugo de Vries. He is from Holland. Okay, and another person, Carl Correns. This person is from Germany. Is from Germany, and one more person is Erich von Techmark, and this person is from Austria. So, okay, these three botanists are rediscoverers of Mendel laws. Right, fine. With this, I will end today's session. So we will see in the next session.